All right, we are here to talk about transcription, and transcription is a process that's going to be converting the DNA code into the RNA code so that DNA and RNA can get that message out to the ribosome. If you remember, the DNA has to stay inside the nucleus. RNA has the ability to leave, so this is a really important part to allow that message to get from the DNA out of the nucleus out to the ribosomes where the protein is going to be made. Now, when we talk about RNA, there's going to be three basic types of RNA. mRNA is the initial copy of the DNA, so the DNA is going to get converted to mRNA during that transcription process. rRNA is short for ribosomal RNA. Ribosomal RNA makes up ribosomes, makes up the actual structure of the ribosomes with some other proteins. tRNA it stands for transfer RNA, and that is going to bring amino acids that are located in the cytoplasm of the cell over to the ribosome to be linked together through peptide bonds and create the actual protein chain. So when we talk about transcription, three things have to happen. One, we have to have a DNA template, and DNA template is just what every cell has. So DNA template's always going to be there. Nucleoside triphosphates, that has to do with, they're called nucleosides instead of nucleotides because of the varying amounts of phosphates in the molecule. So don't worry about that. I don't get into that very much in this class, so that's not that big of a deal in my world. And you also need something called RNA polymerase. Hopefully you look at that name and you can see ASE is an enzyme. This is an enzyme that's going to create the RNA polymer, which is a group of monomers linked together. RNA polymerase is going to work very similarly to the way that DNA polymerase works, in that it's going to take free nucleotides that are inside the nucleus and put them into the form of RNA. So when we talk about transcription, there are three basic stages, and we're going to go through each of these stages now. Just so you have an idea, if we're looking at a cell, we're looking at inside the nucleus. So transcription is only going to happen in this area right here. Outside the cell, that's where translation is going to happen. That's going to be another slideshow. So we're just focused inside the nucleus and that creation of the RNA. The first thing RNA polymerase is going to look for is something called a promoter. A promoter is not a protein. It's not anything crazy. A promoter is just a sequence of bases in the DNA. So if we remember, our DNA has bases A, T, C, G. A always pairs with T, C always pairs with G, and there's going to be some specific kind of sequence inside the DNA strand because our DNA strands are really long. There's thousands upon thousands of base pairs in every strand of DNA. At certain parts, there's going to be something called the promoter. And the promoter basically just tells the RNA polymerase, hey, start here. This is where you're going to go. And on that promoter, there's going to be something called the initiation site where the transcription actually starts to take place. And promoters all vary, and depending on what protein we're trying to make, different promoters are going to be used. So again, a promoter is just a sequence of bases on the DNA that basically are kind of like a signal to the RNA polymerase, start transcribing here so that we can make RNA to make the specific protein. During elongation, the RNA polymerase moves down the DNA template. It moves down the DNA template in the 3 to 5 direction, and the exiting mRNA transcript that's going to come out is going to be in 5 to 3. So it lays it down from the 5' end to the 3' end. So it's very similar to DNA replication. It works basically the same way. Now, when we're talking about RNA, remember our bases change a little bit. We have A for adenine. U is going to be for uracil. That's going to take the place of thymine. So anywhere T would normally pair as in DNA, we're going to pair a U there. So U is always going to pair with A, and then guanine, cytosine, same thing that we had going on there. RNA polymerase does proofread, but it's definitely not as affected. But one of the big deals, one of the good things about RNA polymerase, not a good thing. I mean, obviously we want it to be effective all the time, but it's not as big of a deal if it doesn't work out quite well because the RNA is going to get degraded in a much shorter time. Your DNA you're stuck with for your life. The RNA, that's something that gets used, and eventually after translation, it will get degraded. So if there is an error, it's not going to affect you for the long haul, but it may affect some things, and we'll get into errors on another day. Termination is the end. Transcription is terminated. When that happens, we have a piece of pre-mRNA that's going to be released from the DNA. So now we have a strand of mRNA that's basically as long as the gene that was transcribed. Remember, the transcription started at that promoter, and it kept going until it found something called the stop sequence. And that basically tells it to release the mRNA. Now, if you remember from the last chapter, we found out that a lot of our DNA is kind of junk. It's filled with introns and exons. Introns are the junk segments, exons are the things that actually code for protein. Well, when we code between promoter and terminator, you're going to find both introns and exons. So that pre-mRNA that comes out is going to be as long as that segment from the promoter to the terminator. That RNA is going to get modified, and it's going to get modified by cutting out those introns. The way that those introns get cut out is by something called splicing. Now, this is just sort of a story that's in your textbook. I don't really care too much about it, but basically they found introns by accident. They were looking for things, and they happened to see these loops that showed them that introns were there in the DNA. Again, you can check your book out for that. I'm not going to go crazy on that with you guys. I care more about the process, how they actually found it. There's a picture of the loop right there that we were talking about. That's how they found the introns, because they saw the introns getting cut out, and they were like, oh, there's some junk segments of DNA there that we don't really need. So let's talk about removing. Now, this is important stuff. This process of removing introns is called splicing. We're going to splice the RNA. The way that we splice the RNA is, you can either call it small nuclear ribonucleoprotein particles, or you can call them SNRPs like I do. That's a SNRP right there. That's a SNRP right there. A SNRP is going to attach to each side of the intron. So we have our 5' end of our mRNA here. We got our 3' end down here. There's an exon is the dark green, the intron's the light green, the exon's the other dark green. I see one SNRP at the 5' splice end and one SNRP at the 3' end. And basically those SNRPs are going to come together. When the two SNRPs come together, they create what's called a spliceosome. And a spliceosome is just simply going to cut that segment of mRNA out and remove it. Once it gets removed, chomp, 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 it gets chopped up, and those nucleosides get recycled inside the nucleus. Your body is really good at recycling things, and that's one of the many things that your cells are going to recycle, is those things that were just part of the introns that didn't really mean anything. So now if we look at comparing our pre-mRNA to our mature mRNA, what do you notice? Hopefully you notice that it is a lot shorter, and these are the segments that are actually going to make it out to the ribosome and actually get coded into protein. So remember how we were talking about one gene to one protein wasn't very complex? Then we said, okay, how about one gene for one polypeptide? Okay, well, we're going to modify that a little bit after we talk about how all this stuff works out. Sometimes we have disorders in removing introns or exons, or having some kind of mutations or splicing not going correctly. A common one with that is called beta thalassemia. That is a mutation to the beta globulin inside hemoglobin. Again, we go back to hemoglobin because it's a really easy protein to study. And when that happens, your body doesn't carry oxygen as well. And when you don't carry oxygen quite as effectively as you should, that results in a condition called anemia. So beta thalassemia is just a condition that can cause anemia, and it just shows that sometimes splicing doesn't always go correctly. So we talked about this gene concept modifying itself over time. Well, after seeing that DNA can get spliced and certain parts can come out, and one thing I didn't tell you, sometimes the exons are spliced out too, and sometimes the exons get moved. A better idea of a gene concept would be that alternative splicing is going to result in different mRNAs and different polypeptides, all from a single gene. So we can take one gene and make multiple combinations to get a final mature mRNA. And that final mature mRNA is what's going to be taken out of the nucleus and sent over to the ribosome to go through the process of translation. Now, still not done yet. Inside the nucleus, that matured mRNA still has to get modified a little bit. On the 5' end, there's going to be something called a GCAP added to it. GCAP has some GTP, which hopefully you remember GTP, very similar to ATP, which means it's stored energy. The process of making protein does require some energy, so having that GCAP there is going to provide that energy to help that mature mRNA bind to the ribosome, which a 5' end is going to be the one that binds to the ribosome first. On the 3' end, there's going to be something called a poly A tail. Now, the first time I saw this, I called it a pony A tail, and just don't do that. It sounds dumb. So make sure you call it a poly A tail, which poly means many. There's a lot of adenine nucleotides that are added to the end of this sequence. And that's a good thing because that's going to help protect the mRNA so that it doesn't get digested. Remember, there's a lot of things going on inside the cell. mRNAs are going to get digested eventually. We just want to make sure that some of the proteins are created that we need first. The next step after we have our mature mRNA that has its GCAP and poly A tail is that mRNA is actually going to leave the nucleus through the nuclear pore, go over to the ribosome and create protein. And that's what we're going to talk about in our next slideshow about translation. Hope you enjoyed. I'll see you soon.